Hi, my name is Flávia Petean, and I'll be talking on behalf of my research group, composed by Lei Young, Shannon Corrigan, Sergio Lima, and Gavin Naylor, in a collaboration between the University of Florida and the Federal University of Rio Grande do Norte in Brazil. And the title of our project is The Role of the American Continent on the Diversification of the Stingrays genus Hypanus. But who are these stingrays? Well, they're coastal stingrays, and they're distributed along the American continent, both on the Atlantic and on the Pacific side, as well as one species occurring at the African coast of the Atlantic. And until recently, these stingrays, they were within the genus Dasyatidae, which was a worldwide distributed genus. But in 2016, Last and colleagues, they revised the genus and they resurrected Hypanus and put 8 species within it, based mainly on the mitochondrial gene ND2 and on morphological similarities. However, when we look at these stingrays, we can see that they are varied. So they can have wide distributions or restricted distributions. Some of them can be large specimens like Hypanus americanus, or they can be small like Hypanus mariani. Some of them may tolerate low salinity areas like Hypanus sabinus. Some of them do not tolerate low salinity like Hypanus longus. So we look at these stingrays and a lot of questions pop into our mind, like when did they diversify? What could have happened to them? Why? And we observed that maybe their diversification is related to the American continent as a whole. But what parts of the American continent? So we have to correlate the biology of the stingrays with the barriers that the American continent probably poses to these stingrays and were barriers to their gene flow, to their dispersal. So we're going to raise a few hypotheses. So for example, if you look at the American continent, we can see 4 main barriers. A land barrier, actually 2 land barriers, which are the Isthmus of Panama and the Florida Peninsula, because since these stingrays are coastal stingrays, obviously they cannot cross land, so it is a barrier. And also salinity and sediments like the Amazon River outflow towards the Atlantic, because the stingrays are mainly marine. Few of them are adapted to low salinity areas. So the Amazon River can be a barrier to them. Also depth and distance, because since they are coastal, they cannot cross huge masses of water such as the Atlantic and the Pacific and reach other continents. So they're probably related and restricted to the coast of the American continent. And also the temperature, because we know these stingrays, they do not tolerate low temperature water. So they're restricted to northern and southern hemisphere within the tropical and subtropical temperatures. So these are our hypotheses that I would like to test, but in order for us to test it, we need a robust phylogeny. So we're gonna sequence the whole mitochondrial genomes so we can have more robust phylogeny with more samples, and then we can calibrate our phylogeny and get the divergence dates of the nodes and correlate the divergence dates with the biogeographic events and geological and climatic events to test if they make sense or not, if they correlate or not. So to start, we need to sample. So we sampled all valid species and all putative species of Hypanus, as well as other genera and other subfamilies, so we could delimit the genus Hypanus, and we sampled ourselves, and we also thank our collaborators and colleagues who sent us some samples. Then we did our analysis. So we did a gene capture methodology using an Illumina next generation sequencer to sequence the whole mitochondrial genome. And then we did our analysis on phylogenetic reconstruction, and we calibrated our phylogeny, and then finally we could reconstruct ancestral areas using biogeobursts in R. Well, so this is our phylogeny, and we can notice here, this is the node of Hypanus, that Hypanus is indeed a monophyletic genus. However, in order for it to be monophyletic, we had to include another species, this Phylogenetics genus, that last in 2016, they suggested this species was a Phylogenetics based on morphological similarities, but they did not have genetic samples of it. And now that we could sequence, we noticed that Phylogenetics genus is indeed a Hypanus. So we increased the diversity of the genus by bringing another species to the genus Hypanus. Besides, we also noticed a lot of lineages that were not previously noticed. So for example, we noticed that Hypanus guttatus are in fact two lineages, guttatus and one that we're going to call Affinis guttatus because of its molecular similarity. However, they are not yet described, so we're not going to suggest a new name until more morphology and ecological data are included, but we're going to say they're distinct evolutionary lineages. So what do we have? Well, last proposed 8 species based on NB2 gene. But based on mitochondrial genomes, we identified 14 lineages within the genus Hypanus. So the genus is way more diversified than we thought 4 years ago. And we found out 6 new lineages, and I'm going to say lineages and not species because not all of them are formally described, but one, which is Hypanus bertholletzi, which we recently described. But there are many more lineages, and the genus is very well diversified around the American continent. So let's test our hypothesis on the biogeographic barriers. So we did some analysis, some biogeographic analysis, and the result is this one. So we have our phylogeny calibrated, an ultrametric tree, and the nodes, they're calibrated. And on the tip of the phylogeny, there are the current geographical distributions of each of these lineages. And if we go to the bottom of the tree and we follow the branches, we can see hypothetical and possible ancestral areas of each lineage. And we can correlate these dates to geological and climatic events that happened on the Earth and mainly around and within the American continent. So I'm going to give one example. So we suppose here, I'm going to focus on this part of the tree, on this small plate here. And we can imagine that 10 million years ago, there was only one lineage, this ancestral lineage here, that occurs all on the east coast of the Atlantic, on the American continent. And then this arrow here represents when the Amazon River started to flow towards the Atlantic. And one of the lineages diversified, which was Hypanus mariani here, running away and looking for a refuge in northeastern Brazil in high salinity areas because this species does not tolerate low salinity. Okay, this is one divergence. If you look another one, approximately 4 million years ago, the Isthmus of Panama reached a point in which there was a cessation of deep water exchange. So shallow water was still flowing, but not deep water. And we can say that this was an important event here because probably a population of an ancestral lineage got trapped in the Pacific, and this population could not return to the Atlantic, so there was an interruption of gene flow between Atlantic and Pacific lineages, and which is probably how Hypanus longus diversified around this time in the Pacific and could not return to the Atlantic. And then if we go on time, with the final closure of the Isthmus of Panama, a lot of instabilities on the Atlantic ocean currents, marine currents were a little confusing. And then what happened was one current flowed from the America to Africa around this period, like 1 million years ago. And one population of this green lineage here probably founded a new population on Africa, and they could not return. So these lineages, they diversified because there was a founder event in Africa. So one lineage stayed in Brazil, which is Hypanus bertholletzi, and another in Africa, which is Hypanus rudis. And at the same time, in North America and Central America, another diversification event occurred more recently, probably related to sea level oscillations on the Pleistocene, in which the sea level decreased and the peninsula of Florida was more exposed, creating a huge land barrier, which probably interrupted the gene flow between two different lineages of Hypanus americanus here. However, these Hypanus affinis americanus is still not described. We're going to consider it just as affinis or as Hypanus americanus because it's a very recent event and we still do not have yet morphological characteristics to differentiate these two lineages. But we do know that they are evolutionary distinct lineages. And this is the final result. We could test all of our hypotheses and we could corroborate them all and see that there are mainly four barriers that the American continent poses to these genus of stingrays. So land barriers, salinity and sediments, depth and distance, and temperature. And here is the phylogeny of the genus. So we have a robust phylogeny. Here are all the images of the current valid species of Hypanus and their distributions and their dates of divergence that we could correlate them all to geographical and climatic events within and around the American continent. And that's it. I'd like to thank you the opportunity for talking. I'm open to questions. If you want to email me, feel free to ask any questions or whatever. And I'd like to thank my colleagues, which are Lei, Shannon, Sergio, and Gavin for this amazing project that we have been working on. And of course, I'd like to thank the Systematics Association for this amazing virtual symposium that we have been having here, and also our financial support, Fulbright and Copies and the Florida Museum and the University of Brazil for providing us funding for this project. So that's it. Thank you so much. Just ask me whatever you want.